So welcome everybody, especially welcome to today's engaging webinar. I'm Adila Sayed, Manager of the Optical Biology Core at UC Irvine and core organizer of the Advanced Imaging Methods workshop hosted in Berkeley next week. It's a pleasure to have each of you join today's webinar as we take a deep dive into the realm of cutting-edge imaging, focusing specifically on real-time intravital microscopy and in vivo cellular level imaging in small animals. Let me introduce the distinguished Dr. Pilhan Kim, currently a professor at the Korea Advanced Institute of Science and Technology. Dr. Kim's expertise extends across Graduate School of Medical Science and Engineering, along with nanoscience and technology. He was a research fellow at Harvard Medical School from 2005 to 2010. And in 2017, Dr. Kim took his passion for pushing scientific boundaries to the next level by founding IVIM Technology, a trailblazing company dedicated to pioneering novel intravital microscopy and imaging techniques. If I get the pleasure of having you guys come out to the AIM conference this year, he's going to be there along with some beautiful demos I'm looking forward to seeing. As the talk continues, feel free to share your questions in the Q&A box below. We're here to make sure that all your questions get an answer. And without further ado, I will pass the microphone on to the brilliant Dr. Kim.
Thank you. Yeah, thank you. Thank you very much for the introduction. And then thank you everyone for joining today's webinar. So today I will talk about this real-time intravital microscopy technique for preclinical research. So it's capable of in vivo cellular-level imaging of various internal organs in a live animal model. And then it can also be a very useful tool for the in vivo imaging analysis of drug delivery and drug efficacy, again, in a live animal model. Okay, so maybe some of you already know that the intravital microscopy can provide a live video in cellular level in a live model in vivo, as you can see in the video in the middle. So compared to the X-ray or CT MRI, it can provide a much higher resolution, and then it can also provide a real-time video in cellular level. So to give you an idea, let me first start with this example of the intravital microscopy imaging of the lung of sepsis mouse model. So in this particular example, we stabilize the lung, as you can see in this video. Okay. So to stabilize the motion of the lung, just the part of the lung against this transparent cover glass. So if you look carefully, we have a small hole here, the suction hole. So we can suck out the air from this chamber through this suction tube by using the suction pump. So we can build up the negative air pressure in the value of around 930 to 960 millibar. And that's enough to stabilize the surface of the lung against this transparent cover glass, as you can see in this video. Okay. So after this stabilization, we can do this cellular level imaging from the lung of this mouse model. Okay. So in this example, so we inject FITC dextran through the tail vein intravenously. So this FITC dextran stays in the blood vessel, so we can visualize the blood flow in the lung of this mouse model. And then at the same time, we can also inject antibody conjugate. So in this particular example, we inject anti-LY6G antibody conjugated with Alexa-Fluor 647 intravenously through the tail vein at around 1 hour before the imaging. And that antibody conjugate can level all the intravascular neutrophil, as you can see in this video. And this video is actually 10-minute timeless video. And then it nicely shows that the movement of this neutrophil inside the vessel. Okay. And then as you can see, there are several, maybe 7 or 8 neutrophil make a small cluster in here in just a couple of minutes. And then it physically block this vessel here, right? As you can see now. So, and then this physical blocking of the blood vessel by clustering this neutrophil disturb this microscopic level blood flow in the lung of the sepsis mouse model. Okay. Okay, so let me go back. So this is a very nice example how we can use intravital microscopy to study the cellular-level mechanism of disease progression in the live mouse model in real time. Okay, that was 10-minute time-lapse video, but we can extend our imaging period up to several hours. So this is one example. So in this particular example, again, we are now imaging the lymph node in the live mouse model. So we study the mouse, and then we expose the pathophysiology lymph node by making a skin incision. So in the middle, so we can see this pathophysiology lymph node, and then we put the cover glass on top of it, and then take this video from this mouse model. In this particular example, it's already published. So we inject the T cell and B cell through the tail vein, and then we observe the homing and extravasation of T cell and B cell from the high industrial venue of this pathophysiology lymph nodes. So, and then this is a 2-hour time-lapse video. So as you can see, we can nicely visualize where and when this intravenously injected T cell and B cell extravasate from the this lumen of Henderson Avenue to the lymph node parenchyma. And this arrowhead pointed out the site of this extravasation of T cell and B cell. Okay, so let's, so if you look carefully in this place, so we can see this B cell come in here, okay, and then you now extravasate in these locations. In the later time point, we can also see the extravasation of a T cell at the exact same location. So this 2-hour time-lapse video, demonstrates that this T cell and B cell, the extravasation of this T cell and B cell is very tightly regulated in terms of spatial location along the wall of this high industrial venue. And then this T cell and B cell share this extravasation focus site in the high industrial venue. So again, this is a very nice example how we can use intravital microscope to analyze the movement of the individual cell in vivo microscopic condition in normal physiological level. Okay, so we can nicely see the cell, but in addition to that, we can also see the drug as well with process labeling. So I don't know whether you can see this video nicely, but this is the real-time video obtained from the liver of this mouse model. Okay, so as you can see, so we put the mouse back onto the this heating pad on the stage, and then we make an incision on the skin and peritoneum, expose the liver, and then we put the cover glass on top of it like this. And then we also catheterize the tail vein by using this catheter. And then, in this example, we use type 2 GFP transgenic mouse model. It expresses GFP in all vascular endocellular cell. So in this video, all this green signal is from the liver sinusoid endocellular cell by type 2 GFP. And then through this tail vein catheter, we inject fluorescently labeled nanoparticle. Okay, and then let me go back. So, and then this video starts when we did the intravenous injections. So as you can see in this video, after around 10 seconds, we can see the arrival of this intravenously injected nanoparticle inside the liver. Okay, then so we can nicely distinguish the individual nanoparticle flowing through this liver sinusoid, and then at the same time we can also nicely identify where this nanoparticle, intravenously injected nanoparticle, started to accumulate inside the liver sinusoid, as you can see in here, and also in here, and then there are several other areas in here as well. Okay, so this is a very nice example that we can use intravital microscope to monitor the delivery of the intravenously injected drug. In this case, it's a nanoparticle. But in addition to that, we have been able to image the antibody drugs or peptide or exosome drugs, and then other microvesicles as well with the process labeling by using our intravital microscope system. So that was a real-time observation, but we can extend our imaging period up to 24 hours, as you can see in this particular example. This is also published work. And to image the same location for this long period of time, we use the imaging chamber called dorsal skin fold chamber, as you can see in this photo. So this is dorsal skin fold chamber can be implanted on the back skin, the dorsal skin of this nude mouse model. And then we inject the triple negative breast cancer cell, MDA-MB231 GFP cell, in the middle of this dorsal skin fold chamber. And then we let the mouse to, and then we let this injected tumor cell grow for 2 to 3 weeks. And then we anesthetize this mouse and then take this kind of images in the middle of this MDA-MB231 tumors in these dorsal skin fold chambers. And then this is 2 hours and 6 hours and 24 hours after the intravenous injection of this nanoparticle, this QAMP Alexa-Fluor 555. Okay. And then this green signal is the triple negative breast cancer cell, MDA-MB231 GFP. And then the blue color, it shows the vessel in this tumor. And in this example, we used antibody conjugate, anti-CD31 antibody conjugated with Alexa-Fluor 647. So let me remove the green color here. Okay, so you can, by just looking at the vessel, we can identify the same location at different time point, like 2, 6, and 24 hours. Okay, so this is exactly same location. And then we can, not easily, but we can find out the same location inside this dorsal skin fold chamber by looking at the vessel structures. Okay, so this is exactly the same location. And then the red color is this nanoparticle delivered to this MDA-MB231 tumors. Okay, so at 2 hours, this nanoparticle mostly stays inside the vessel, like this. At 6 hours, we can see the increase of this delivered nanoparticle, but still mostly inside the vessel. And then finally, at 24 hours, we can see this nanoparticle now get out from the vessel and then deliver to the tumor cell, as you can see in here, and also in here, and then some in here. Okay, so this is the good example how we can use intravital microscope to observe the delivery of the drug to the tumor cell for extended period of time, like 24 hours. But we can extend the time period even more by using this window chamber, and then we can find the exact same location like this example in this slide. Okay, I started with by showing several examples of intravital imaging study. Okay, to give you the idea how we can use intravital microscope to study the cellular-level biology in the live mouse model. So intravital microscopy is a microscopic scale imaging technique, and then it can analyze the 3D cellular-level movement in a live mouse model. And this IBIM technologies, so our intravital microscope system can enable 3D cellular-level imaging in the live mouse model. So we can visualize the cell trafficking, as you can see in these videos, and then also we can see the cell-to-cell or cell-to-microenvironment interaction in a live preclinical animal model in vivo for a couple of hours to a couple of days to a couple of weeks. And also, as I showed you in this example, for the drug development, this intravital microscopy also can be a very useful tool because it can enable direct in vivo imaging analysis of a drug delivery to the target cell or target tissue. And then later I can show that the intravital microscopy can also do monitoring, in vivo monitoring of the drug efficacy, and to validate, and then also it can be utilized to validate the mode of action of that drug, again, in a live clinical animal model in vivo. And then we have been able to visualize almost every organ and tissue in a live mouse model. So for each specific organ, we have our own accessories and imaging devices, imaging window device to visualize all these different organs in a live mouse model. So all these images were obtained from the live anesthetized mouse model by using our intravital microscope system. We have been able to image the bone marrow in a live mouse model, as you can see here, brain and brain tumor, or retina in the eye. This is the skin, and then hair follicle in the skin, again, from the live mouse model. And then we have imaged the various tumor model. In this particular example, this is the colorectal cancer in the CCAM. And then we have been able to image the pancreas, spleen, gastrointestinal tract, like a stomach, small intestine, colon. We have imaged the various adipose tissue all around the body, prostate, placenta, and the lymph node, the memory tissue, and cell transplant. In this green signal is the transplanted cell. Kidney, muscle, liver, heart, lung, and so on. So the list of the organs is keep increasing by using our intravital microscopy system. And then again, I'd like to emphasize that all these images were obtained from the live anesthetized mouse model in vivo. Okay, so let me spend some time to introduce the key feature of our intravital microscope system. So this is IBM CMS3 model, it's a dual mode, intravital confocal and two-photon microscope system. This is a very much optimized system for in vivo cellular-level intravital imaging of a live animal model, as I showed in previous slides. And then it has all-in-one packaging. So everything is inside this single box. Okay, so it's easily installed in anywhere inside the lab or even in the animal facility. And then inside this box, it's capable of 4-channel simultaneous two-photon imaging with a compact 920 nanometer femtosecond pulse laser module inside this box. In addition to that, we can also connect the conventional tunable titanium sapphire femtosecond pulse laser for two-photon imaging as well. So it has additional optical ports behind this box, at the back of this box, so we can easily connect the conventional laser system as well. And then also it can connect with the fiber-coupled 4-wavelength CW laser module for 4-channel confocal imaging. So this microscope is capable of both confocal and two-photon imaging with 4 channels. Okay, let me show you just these short videos. So as sorry. So as I explained to you already, so we can connect the conventional titanium sapphire laser like this, okay? So this is the conventional titanium sapphire laser we can connect through this mounting system, laser delivery system, delivery modules. Okay, so this is a single box, so you can open up this sliding door in front, and then we can see this objective lenses inside. And then inside the stage, this is the heating temperature control stages. So we can put the mouse inside like that. And then we just close the door. So you don't need a dark room to use this microscope in your lab or in the animal facility. And then we can obtain the cellular videos. So this is confocal laser beam path. And then as I told you, so we have a 4-channel confocal detectors inside. And then this is the fixed wavelength two-photon lasers, femtosecond lasers, or we can connect the external laser system as well. And then we have a 4-channel NDD detector for two-photon imagings. Okay, so I think most of you already know what is a confocal microscope, but I just want to explain very simply. So confocal microscope is a technique that can obtain the optical section image from the sick tissue by using the pinhole in front of the confocal pinhole in front of the detector, as illustrated in these figures. Okay, so I just showed you the one example of this confocal microscope, intravital confocal microscope imaging obtained from the inguinal antibody tissue of this mouse model. in vivo. So in this particular example, we use adiponectin-free MTMG mouse model. So all the adipocytes express membrane GFP. So in green color, we can see the adipocyte. And then we inject another fluorophore, SF44, intravenously, and then this SF44 label the lipid in the live mouse model after intravenous injection. So you can see the lipid inside this adipocyte in green colors. And then all other cells express membrane TD tomato. So we can see the vessel and then some immune cells, in the perivascular immune cells like here. And then as you can see, by using confocal microscope, we can obtain the G-stack images with optical sections with 4 micrometer interval, okay? In this thick inguinal adipose tissue. And then later, of course, we can make this 3D rendered video. And then we can nicely identify the 3D subcellular level structure from this G-stack imaging data, as you can see in this video. Okay, and then in addition to confocal microscope, we can also use two-photon microscope for in vivo intravital imaging of live mouse model. And then in case of two-photon microscope, by using longer wavelengths near-infrared femtosecond pulse laser, we can achieve deeper tissue imaging with optical sections. Okay, and then in addition to the process imaging, the two-photon microscope is also capable of multi-harmonic generation imaging without any labeling, like second harmonic generation, SHG. And then this second harmonic generation imaging is particularly useful to visualize the collagen in the live tissue in a live mouse model. So this is one example. This is again the in vivo imaging data obtained from the liver fibrosis mouse model. So in addition to the fluorescence signal of repeated droplet in this mouse model in the liver, it's labeled by SF44 again. So this red color is the lipid in this liver tissue in a live mouse model. And the green color is the collagen, the fibrotic collagen visualized by the second harmonization signal with the two-photon microscope. Okay, so from now on, let me just quickly show what you can expect to obtain from the live tissue by using our two-photon system. So this is the example of in vivo 4-channel two-photon imaging by using this 920 nanometer femtosecond pulse laser inside this microscope box. So this is maximum intensity projection images obtained from the skin. And this is in this example, the white color is second harmonization signal. It's mostly collagen. And then we image the H2B GFP. So every cell's nuclei express GFP. And then we use MTMG mouse model. So all the cells membranes express TD tomato. So in red colors. And then we also inject another fluorophore EB through the tail vein. So it labeled the blood vessel in this mouse model. And then this is the G-stack data, like this. So if you are familiar with the micro anatomy of the skin, so you can now see so this is the very outside between the epidermis and dermis. And then if we go deeper, we can see this very strong second harmonic generation signal from the dermal collagens. And then we can start to see these hair follicles in here. And then this hair follicle is like this. And then we can see the capillary in the dermis. And then we can see this big vessel in deeper area of the skin. Okay, and then this is images obtained from the muscle, again, in the live mouse model. So in this case, this white color is from the fascia, the very outside of the muscle. And then if I go deeper, we can see this myocyte. And then at the same time, we can also see the sarcomere, the myofilament by using the second generation signal. So you can nicely see in white color the second generation signal from the myofilament in the sarcomeres. And then the green signal again, the nuclei of the myocyte, and then MTMG from the membrane of these myocytes. And then we can nicely see this big vessel in blue colors, it's pseudo colors. And this is the pancreas. So again, we can nicely see the subcellular level structure of the pancreas in 4 colors by using our 2-photon microscope. And then this is from the kidney. We can nicely see the tubular structure of the kidney. And then we can identify all these cell nuclei. And then this is another example, just spleens. Okay, and then we also have our own visualization software. So we can capable of the 3D imaging and 3D rendering. And also by taking a timeless 3D geostat data, we can visualize it in 4D like this. So we can nicely see the cell movement in 3D space in the live tissue in a live mouse model, like this. Okay, so there are another picture. So this microscope is equipped with very high-speed laser scanner for real-time imaging with the speed of 30 frames per second to 100 frames per second with 512 by 512 pixels per frames by using rotation polygon meters. So it can achieve very high speed. And then this 30 frames per second is fast enough to see the circulating cell in the vessel, like this example shown in this video. So this is again, this is real-time video obtained from this total skin ball chamber mouse model. So in this particular example, we inject this LSGFP cell in the middle of this total skin ball chamber, and then we just pseudo-color in blue color. So this blue color is the LSGFP tumor cell. And then after 2 weeks, we take out the small amount of the blood, and then we label it by using fluorophore DID, and then we pseudo-color it in green color in here. And then we inject the anti-CD31 antibody conjugate together with this fluorescent red blood cell and then the CD31 antibody conjugate, fluorescently labeled the endocellular cell, as you can see in these videos. So red color, we can see the vessel endocellular cell. Green color, we can see the flowing red blood cell. And the blue color is the tumor cell. Okay, so we can, so this is a good example, we can utilize our real-time imaging capability to analyze, to visualize the flowing cell in the tumor vessel. Well, this is another example. So I already explained this device for lung imaging. So this is the real-time video, again showing the flowing red blood cell in the capillary of this lung of this mouse model. And then even you can image the beating heart as well. So again, we use this simple device to stabilize the beating heart surface motions, and then we can obtain this real-time video from the surface of this heart. Okay, and then very recently, we, in collaboration with another professor in KAIST, it's not published in Nature Methods, we developed the AI framework to effectively reduce the noise from the real-time video. So on the right-hand side, this is the real-time video, the raw video obtained from the skin of this mouse model. And then we inject the red blood cell, and then it's pseudo-colored in blue colors. So it's very nice, but still we can further improve the signal-to-noise ratio by applying our AI framework. And then we can very nicely denoise our real-time video, as you can see in this example. This is another example obtained from the muscle. So we can nicely see the real-time phenomena, such as flowing red blood cell, with much less noise by using our AI method, processing method. This is another example obtained from the kidney of this mouse model. Okay, and then in addition to this real-time imaging capability, And then it also helps us to compensate the tissue motion very nicely. So like this. So our approach is very straightforward. So we first obtain this real-time video rate imaging from the moving tissue in the live mouse model. And then we can do frame-by-frame image registration to compensate this cellular-level tissue motion. Like this, okay? So to do this kind of motion compensation, you need two things. First, you need very high-speed imaging, like at least 30 frames per second, like this. And then you need this automatic image registration to compensate the motions. And then you can just compare these two images with motion compensation or without motion compensation. So as you can easily imagine, without proper motion compensation, you will always end up with very blurred images due to motion artifact. So this is another example. So this kind of tissue motion can be very nicely compensated by using our microscope. So this is with or without motion compensation function. So by applying our motion compensation function, we can nicely achieve subcellular level resolution in the moving tissue, like this in this example. And then we make this motion compensation function can be performed automatically. So this is our software UI, user interface. And then what you need to do is you just turn on the motion compensation function. And this is just a click the button. And then it automatically compensates the motion compensation from this kind of movement can be compensated. And then it can give you this compensate, and then it can generate this motion compensate the subcellular level images, as you can see in here. Okay. So, and it's all, full automatic. You just need to just one click, and then this, motion compensation can be done, automatically. Okay. And then we also provide this, animal body temperature monitoring and then maintaining system. So this is a touchpad tablet for live animal temperature monitoring. So we normally use this kind of electron probe. It can be inserted through the anus, and then it monitor the body core temperature. And then we feedback control our heating plate to keep the mouse body temperature to be stable at 37 degrees after anesthetization. And then so we can keep the body temperature of this mouse model for very long time, like during our intravital imagings. And then we also provide our these glass heaters to keep the glass temperature also to be 37 degrees during covering the exposed tissue like this. Okay. And then when you open up this sliding door in front of our microscope box, this stage, motorized stage, also have a sliding mechanism like this. And then the heating plate can be easily detached or attached like this. And then we put our microscope in the stage like this, and then close the door. And then we can also connect the gas sanitization system, very conventional, anesthetization system using a spray like this. So this is our microscope. So we have this side window, and then we have a tube adapter here. So we can connect the tube from the vaporizer of isoprene through these gas flow meters. And then we can connect it to the mouse model like this by using our head mask. So we can keep the mouse to be anesthetized as long as we want by using this isoprene-based gas sanitization system. Okay, so, and then now, I already spent more than 30 minutes. So from now on, I just briefly show how we image all these different organs in a live mouse model. So yeah, this is one example of skin. So this is ear skin imaging. So we have this U-shaped bracket with the transparent cover glass in between in here. So we simply attach the ear skin to the transparent cover glass like this. Okay, and then with the motion compensation functions, we can obtain this kind of a timeless video from the live mouse model. So in this particular example, we use Rhizozyme GFP mouse model So we are imaging the neutrophil and the macrophage in the skin. And then we also label the endocellular cell, again, by injecting anti-CD31 antibody conjugate intravenously at 1 hour before the imagings. This is one of our work published in JCB. So in this particular example, what we did was we inject quercetin labeled protein by intradermally by using microinjector, and then we observed the cellular-level response at 3 hours and 6 hours after the injection of these proteins. So we can nicely see the recruitment of this neutrophil and macrophage to this injection site of the proteins. And then this is our observation of the bone marrow after bone marrow cell transplantations. So for bone marrow imaging, we normally image the cranial bone like this. So just make a small, just a skin incision on the head, and then we can expose the cranium like this. And then from this cranium, we just image the very wide area by taking TIE scan images like this. And then we can repeat this kind of wide area imaging in a single mouse model every day from day 1 to more than that. So this is the same mouse model. Again, we use vessel labeling to find out the same location. So we can see the everyday changes in cellular-level after this bone marrow transplantation. And then you can nicely see this from day 3 to day 4. We can see the very dramatic increase of the transplanted cell, like this from here to here. And then if we take a timeless video of this one side, this is what happens. So we can see this proliferation of the cell in here, and then later in here also as well. Okay. Even we can extend our imaging field, more than 10 days. So this is from day 2 to day 10. Again, by labeling the vessel, we can identify the same location in this mouse model. So this is longitudinal, repetitive, wide area imaging of the cranial bone marrow after bone marrow transplantation of this LSK bone marrow cell. So as you can see here at day 3, we start to see this, just couple of cells, and then it very rapidly, proliferate and then peel up the this bone marrow sinus like this until day 7. And then at day 7, this proliferated bone marrow cell mobilized and they go to the periphery. So it empties up there like this. And then we can see the neighboring area, started to proliferate with this bone marrow cell, transplanted bone marrow cell. So this is a very nice example what you can do by using repetitive, longitudinal intravital microscopy in cellular-level. So this is the liver imaging. So I already explained. So let me just go on. So, and then this is one example, another example of the liver imagings in longitudinal manner, 2 days with MCD diet to induce the fatty liver, 7 days, and then 14 days. So in this particular example, we injected this Pro4 SF44 intravenously through the tail vein, and this SF44 specifically labeled the lipid in whole body. So we can nicely see the lipid droplet in the liver of this mouse model. So it's like the 2 days, 7 days, 14, and 21 days. And then at every time point, we label the lipid droplet by intravenously injecting this SF44. And then at the same time, we also label the vessel by injecting this CD31 antibody conjugate. And then this is another example. So in this case, we visualize the collagen in the liver fibrosis model by using a two-photon microscope. So this green signal, green color, is perisinusoidal collagen accumulated in this liver fibrosis mouse model. And that color shows the hepatic lipid droplet. And then we can also image the small intestine. This is what published almost 10 years ago now. So this is the transport of the fluorescent PET from the small intestinal lumen to the mesenteric lymphatic vessel. from 0 minute to for 26 minutes. So you can see the transport of this fatty acid maximized, peaked at 90 minutes after the application on the luminous side of the small intestine. And then there are various imaging window technique for longitudinal EPTD imaging. So this is those skin fold chambers. I already showed you. So this is our kind of educational video to quickly show you the procedure of the skin fold imaging chamber implantation, like this. Okay, let me just quickly go on. So once we make up this window on the back skin, in the middle, we can inject the normally press the GFP expressing tumor cell in the middle of the dorsal skin fold chamber, like this. And then we seal up this dorsal skin fold chamber, like this. This is the sealing to keep the glass. And then we can use our intravital microscope to image this dorsal skin fold chamber repeatedly in this mouse model. So this one I already explained. So this is I already explained. So we can image the drug delivery to the tumor cell like this by using our dorsal skin fold chamber technique. Or we can, even have a more longer time interval, like a 3 days interval observation of the tumor vessel in this mouse model. So like 7 days, 10 days, and 13 days after the tumor cell implantation. And then we can see the change of the growth of this tumor cell together with the change of the tumor vessel, like this. And then we can also compare the change of the vessel remodeling with and without the anti-angiogenic treatment. So you can nicely see the efficacy of this anti-angiogenic treatment in the mouse model. And then we can image the exact same locations like this. And then there's abdominal imaging windows, also optimized for our microscope. And then we can implant this imaging window to visualize the abdominal organs like a liver, kidney, pancreas, spleen, and so on for a couple of weeks to 1 month. Let me show you. So this is just one example. So in this particular example, we implant this abdominal imaging window on the side of the abdomen of this mouse model. So this is the pancreas, this is spleen, and then we inject this pancreatic tumor cell in the middle of this into the pancreas, and then we can image the changes, the growth of this tumor cell in the pancreas from day 9 to day 14 every day, and then we can also image the tumor vessel using CD31 antibody conjugate. So you can nicely see the proliferation, the growth of this tumor cell together with tumor vessel remodeling, like this. And the cranial imaging window, it's also very well established technique. So we remove the small part of the cranial bone using the drill, and then we put the glass on the exposed area, like this. And then after this surgery, we can keep the mouse model for several months, and then we can repeat the brain imagings. So this is the wide area images after these surgeries. So through this glass, through this cranial imaging window, we can spare the meninges on the surface of the brain. So this is the G-stack video. So we can see the meninges, we can see the vessel on the top surface of the brain, and then we can see the brain cortex, like this. So you can now see this penetrating vessel and the brain and then cerebral cortex. And then this is another work published in Glia. And then in this particular example, we make a small impaction in the penetrating arteriole, and then we observe for 1 month, and then to see the cellular level changes. And then this is repeated images of the brain cortex from day 0 to day 5 after this impaction of the single penetrating arteriole in the brain. And then we can see this vessel leakage, suggesting the BBB dysfunction with vascular leakage at day 3. So if you can see this red color, this is 3 kilodalton PRIT stretch dextran, and then you can see the leakage at day 3. But it's spontaneously curved at day 5, like this. So by just looking at the vessel shape, we can identify the same location, and then we can see this transient, BBB dysfunction. And then we observe the parasite like this for 24 hours. So we anesthetize we keep the mouse to be anesthetized for continuously for 24 hours, and then we can see this recovery of parasite and vessel coverage like this. So we can see this elongation of parasite processes along the vessel like this. This is what happened during this recovery of the BBB dysfunction from day 3 to day 5. So this is from day 3.5 to 4.5. So this is the cellular-level mechanism underlying this recovery of the BBB dysfunctions in this mouse model. Okay, I just quickly show you how the example of the intravital imaging of all these different organs. So if you want to learn about more about our intravital microscope system, please visit our webpage www.ivimtech.com, or just send us an email to information@ivimtech.com. So we have specifications, and then we have also another customization option depending on your application needs. And then we also have a lot of information about what kind of disease model and what kind of quantitative analysis you can do by using intravital microscope. And then we have uploaded a series of webinars on our webpage with specific topics, like exosome imaging or peptide imaging or GI tract imaging, or more universal general talks about the in vivo imagings. And then we also provide the research service as well. So we have our in-house animal facility and in-house imaging lab. So if you can just ask us to do intravital imaging experiment by using our in-house facility and then our intravital microscope. So this was an example we have done to visualize the drug delivery. So this is exosome. This is the red color is the exosome, like 10 minutes and 30 minutes after the exosome injection through the tail bed. So we can see the delivery of this exosome to the target cell and then internalization. And this is antibody drug delivery monitoring through the BBB in the neurodegenerative disease model in the brain. Okay, so IBIM technology, we are specialized in this in vivo comfort core and 2-photon all-in-one intravital microscopy system. It's capable of automatic motion compensation function to enable all different organ imaging inside the live mouse model. And then we also provide intravital imaging research service. And then we have a specialist team with very deep know-how of the intravital imaging with more than 10 years of experience. So we can provide very in-depth consulting or technical support to design this intravital imaging experiment, and then to help you to do your own research. And then we also provide the training service for the surgical skills to do this kind of intravital imaging studies. Okay, so thank you very much for your attention. And then we will have another chance in the AIM workshop in next week, I think. Thank you very much.
Great talk. Thank you, Dr. Kim. That was incredibly informative. And I want to especially say your videos showing how to actually mount the animal and image with the animal were very, very helpful. It really helps make it clear. I open it up for questions, the Q&A. Happy to read those for you. So First one is, how do you mitigate the impact of lipofuscin
Mhm.
autofluorescence, especially in the older samples, especially in the brain, where you know you get a lot of autofluorescence in the green channel?
Okay. Okay, this is a very specific questions. I don't have a much experience in aged brain sample. Uh, so, but we have a we can try like a general technique such as unmixing spectral unmixing technique. So we can do sequential, imaging with different laser wavelengths, and then we can do spectral. And then we can apply the spectral unmixing to mitigate the autofluorescence signals. So
Is
it's kind of very standard, method.
okay. What about I'll just ask it on my own,
Oh.
any kind of lifetime imaging, fluorescence lifetime imaging.
Mm-hmm. Uh, yeah, lifetime
Especially
image.
since you're coming over to AIM, right? There's a
Mm-hmm.
lot of limb imaging and
Mm-hmm.
expertise at
Mm-hmm.
the AIM conference.
Yeah, sure. Yeah, Plim can be also solutions, I believe. And I have not I have not tried Plim imaging in the live tissue by myself, but we have another person in my company uh, who are who have experience in Plim imaging with intervital confocal microscope and two photon microscope. So he may be a better person to answer the question about the plea.
Sounds good.
Okay.
Another question is, how easy is it to take multiple fields of view at one time point from the same mouse and then go back and recapture the same fields of view at another time?
Okay.
So kind of
Well,
like
so
multiple
yeah,
positions.
okay, yeah. So for repeated imaging, but let me go back. I'm sorry, yeah, it's a little slow. So it's not too difficult, So with imaging windows, so with the imaging windows, we can already know where to image it in gross scale. For example, let me go back. Yeah, for this example, right? So in this
Mhm.
case of the pancreas imaging, for example, so we already know, where to image it roughly. And then we always use the vessel as our, anatomical landmark to find out the same locations. So in our microscope, we first start with the low magnification objective lens to identify the where to look at. So we just identify the big vessel as our
Mhm.
anatomical landmark, and then we follow it. We use the very big vessel as our landmark to find the microscopy location. And then to do that, we use our low magnification objective lens. And then we change it to high magnification objective lens. And then we are able to find the exact same location like this, shown in this slide. So we have so low magnification objective lens and high magnification objective lens, we can switch it automatically with the motorized turret system, and then just label, and then when we label the blood vessel, it's not difficult at all, I say.
Okay, nice.
Mhm.
Great. Another question is, so
Yeah.
your AI processing
Mm-hmm.
they're asking if that's
Yeah, yeah.
post-acquisition or real-time during data. I mean, that's
Mm-hmm.
beautiful processing. It
Mm-hmm.
looks really just like you took a
Uh,
nice slow image.
yes, yeah, it's post acquisition actually. So
Okay.
yeah, it's post acquisition processing. So we first acquire this raw video, and then we do post processing to remove the
And
noise.
I
So
don't
it's
remember if you mentioned
mhm.
what is the size of your data files here?
In this case, well,
Like a 2 minute, 10-minute video? Are we talking terabyte,
no,
gigabyte?
no, yeah, it's I think this is maybe uh, 1 minute video will be several hundreds gigabytes, I believe. Yeah. So it's well, it's
And
in the
nowadays,
range of
that's manageable
several.
nowadays.
Yeah. Yeah,
Yeah,
it's manageable nowadays. Yeah.
yeah. And then another questions from Philip again. Any technical difference between the IVM CM3 and the IVM MS3?
Okay. So in case of, okay, let me go back. Yeah, maybe I should show it. So it's basically same. So it use the same detector, but when we say CM3, it means we use this external, conventional titanium sapphire lasers. That's for the CM3. But in case of MS3, we use fixed wavelength laser inside this microscope. So that's the only difference. So that's the only difference. Whether we will connect the external laser, like a titanium sapphire pentasecond pulse laser, or use the integrated fixed wavelength 920 nanometer pentasecond pulse laser. That's for the MS3.
Okay.
Mm-hmm.
Any other questions?
Okay. So
one more. What is the type of file formats?
Well, any common image format we use. So of course we use our own file system, but you can export it to like TIFF or JPG or AVI videos or MPG videos.
Okay.
So we have an export protocol.
Okay. Any other questions? All right. Well, as we wrap up, I just want to point out that we're going to be happy to host IVIM over at the Advanced Imaging Methods Workshop coming up not in about 9 days. So we're excited to have you guys over there, and they're going to be presenting again on their imaging session Thursday morning, and we'll also be at the vendor booth fair Tuesday through Thursday. So for all of us showing up in person, I know we're excited to see what you guys have to play with your toys while you are at AIM. So
Great.
thank you everyone for being a part of this talk, and Dr. Kim, if there's any final words, if not, I think we can wrap
Okay.
up.
Yeah. Thank you very much for attending. And then, so as like the side mentioned, we will physically attend the AIM workshop, and then I will be there as well. So if you have any more questions, if you have any question, happens in your mind, and then please come to me, our booth, and then ask me. Thank you.